All right, so now we're making our way down to the small intestine down here. But just to kind of recap what happens, so we've ingested food into the oral cavity. What happens there? Okay, a lot of mechanical breakdown happens there, from the tongue, from the teeth. Any other digestive activities happen there? What about some digestion? What type of digestion occurs in the oral cavity? Yeah, that's where we get these salivary glands secreting saliva with the enzyme amylase, and that starts the breakdown of carbohydrates. Okay, so all that happens, and then we swallow that food, which is now called a bolus, and it goes back to this next section here called the pharynx. Then it travels from the pharynx down through the thoracic cavity via the esophagus here. And what's the type of movement that's transporting this food bolus down through the esophagus and also through the entire digestive tract? And the answer to that is peristalsis. So we're moving that food bolus down through the thoracic cavity, through the esophagus. Now it punctures through the diaphragm, and it gets into the stomach. And this is where a lot of activity starts happening. Okay, we start mixing stuff together, mechanically breaking it down. We also get the digestion. We also start the digestion of another type of nutrient. And what type of nutrient is that? Those are proteins. And so now we go from the stomach then into the small intestine. And the small intestine has three components to it. Here's the stomach, and the first part of the small intestine that comes right out of the stomach is called the duodenum. And I'll show these portions in a little bit better detail here. We have the duodenum, then we have the jejunum, and then we have the last part, the ileum. And then the ileum takes that material, which is now going to be called chyme, and delivers that to the large intestine, in particular to the cecum portion of the large intestine. Okay, but the small intestine though, what does the small intestine do? No big deal, but it just happens to be the body's major digestive and absorptive organ. So pretty much all digestion is going to be completed at the small intestine, breaking those nutrients down into small enough components where they can move into the blood. So not only is digestion going to be completed here for the most part, but virtually all absorption is going to occur at the small intestine. So taking all those nutrients in their smallest components and moving them into the bloodstream so they can get into the body. So like I mentioned, the small intestine is divided into three regions. In the first region here, maybe I should draw this. This is where the stomach would be. The first region is called the duodenum. And this is the duodenum right here. It's the shortest part. It's only about 10 inches long. And it's shaped in a C. It has a C shape to it. Now something I wish was on here that happens to be a handy identification marker is the pancreas. So this duodenum is in a C, and the head of the pancreas sits right in the C of that duodenum, and then the pancreas is kind of a tadpole shaped, does something like that. But that's where the pancreas sits. And this is handy for a couple reasons. If you're just trying to orient yourself, you'll say you're looking at a cadaver and you're trying to find either the duodenum or the pancreas. Well, if you find the pancreas and the pancreas is this glandular looking tissue, then what that means is that you can identify the duodenum because it sits right there in the C of the duodenum. Likewise, if you find the duodenum, then you're able to find the pancreas. The duodenum is what we call the mixing bowl. Okay, we have a salad here, and we're mixing all these components of the salad together: salad, vegetables, dressing, all this kind of good stuff, croutons, not for me, but all that kind of good stuff, mixing that all together. And the same thing can be said about the duodenum. So here we have the duodenum, this is the bowl, and we're going to mix a whole bunch of stuff in there. So what's located in the duodenum? Well, one, it receives it receives chyme from the stomach. So after that food bolus gets mixed around in the stomach, it's now called chyme, and that chyme gets delivered, if this is the stomach right there, the chyme gets delivered into the duodenum. So that's one thing that's in there, is you have chyme. But what else? And this is where we can look at this figure to kind of deduce this. So right here, we have an opening into the duodenum. And then you can see there are these ducts that potentially secrete things through that opening into the duodenum. So let's take a look at, first of all, let's see. Let's go up here. This right here is called the bile duct. And where's the bile duct going? The bile duct makes its way up into the liver. So the liver's going to secrete a digestive product that's going to travel down the bile duct, and it's going to be secreted into the duodenum. Now what is that digestive product that the liver's producing that's secreted down there? And the name of the duct gives that away. It's bile. So the liver's going to produce bile that's going to make its way down into the duodenum, and we'll talk about what bile does. Now, any bile, you might be saying, oh, what about the gallbladder? I thought the gallbladder made bile. Well, the gallbladder does not make bile. Instead, what it does is it stores bile. So any excess bile that's made that doesn't go into the duodenum here gets backed up, and it gets stored in the gallbladder, which is handy. So we have bile from the liver and the gallbladder. Then let's follow this duct right there. That duct is called the pancreatic duct. And the pancreas has a tremendously large role with regards to digestion. And what the pancreas is going to do is it's going to make and secrete digestive enzymes into the pancreatic duct. Then those enzymes are going to travel down the pancreatic duct and be secreted into the duodenum. So we have digestive enzymes from the pancreas, and we also have bile from the liver that are mixing together with that chyme, with the goal of completing digestion and allowing for absorption. All right, so we have all this material in there, and we have to mix it together. And so, how are we going to mix it together? First of all, I guess, let's see. Well, let's just go here. So here we have chyme that's in the small intestine. And the goal is to mix this all together so we have this nice homogeneous mixture in there. We're not only mixing the chyme together with each other, but we're mixing it together with the bile and the digestive enzymes. And the type of movement within the digestive tract that carries that out is called segmentation. There are these rhythmic constrictions of the small intestine that, like this says, it mixes food with digestive juices, and it also increases the efficiency of absorption by moving this food mass repeatedly against the wall of the small intestine. So this kind of looks similar to peristalsis, except peristalsis, the main goal is just to move the material down through the GI tract. Segmentation, we're actually trying to move it together. But they do have some commonalities between the two. What layer of the small intestine is going to be responsible for this segmentation? The muscularis layer, okay, the muscularis externa, just like peristalsis. And just like peristalsis, this is going to be regulated by the enteric nervous system. So we hit the wall of the GI tract detects stretch, then it works back and causes these constrictions because of it. And we're going to find segmentation not only in the small intestine, but the large intestine as well. The next place that we get to is the jejunum. And so you have what's called this duodenojejunal flexure, which I'm not concerned that you know about necessarily, but it does mark the boundary between the duodenum and the next part, the jejunum here. And the jejunum's longer. It's about 8 feet long. And this is where the majority of digestion and absorption are going to occur, is in the jejunum. So we mixed all this material together here in the duodenum. Now it moves down into the jejunum, and this is where the final digestion and absorption can take place. And so that's what these figures are showing here. Here's a complex carbohydrate, and what we're going to do here is we're going to digest it into individual monosaccharides. Here is a protein here, and we're going to finish digesting it into individual amino acids. And then after that digestion can occur, that allows for those monosaccharides to be absorbed into the bloodstream, for these amino acids to be absorbed into the bloodstream. And then the next part we have is the ileum. The ileum is about 12 feet in length. And what's going to happen in the ileum? Well, we're going to have vitamin B12 absorption here. We'll talk, well, we talked about vitamin B12 in the stomach. Okay, you need that intrinsic factor being produced, so it allows vitamin B12 to be absorbed. Well, it's going to be absorbed specifically in this ileum region here. And then also any other products not absorbed by the jejunum are absorbed in the ileum here. So here we have the ileum. It's about 12 feet in length, and eventually it's going to make its way and deposit its products into the large intestine. This is the large intestine, the first part of the large intestine there, and in particular this region right there is called the cecum. So we have materials from the ileum going in to the cecum. Now, not surprisingly, there's going to be a sphincter that separates these two, that's going to regulate the passage of material from the ileum into the cecum. And that's what we're looking at over here. Here we have the ileum, there's the ileum, and that's going to deposit its products into the cecum right there. And right there is the sphincter that separates it. And what's that sphincter called? Well, it's called a valve, but it's actually a sphincter. But more importantly, it's called the ileocecal valve. Ileo because we're dealing with the ileum, and cecum because we're dealing with the cecum here too. So it's the ileocecal valve. But again, it's a sphincter. It's a modified layer of the inner circular layer of the muscularis externa. Alright, so not surprisingly, the small intestine is highly adapted for nutrient absorption. Nutrient absorption is its function, so it has to have the appropriate structure to allow for that. Complementarity of structure and function. And let's go through some of these here. First of all, this is the small intestine here. Okay, and as you guys saw, the length alone of the intestine provides a huge surface area. About 20 feet long the intestine is. So you have a huge surface area just for this digestion and absorption to occur in. And then it also has 3 structural modifications within the wall itself that greatly amplify its absorptive surface area and increase absorption efficiency. So let's look at these 3 modifications. Well, I guess I'll show you them right here. The first thing are called these circular folds. So the lining of the small intestine isn't necessarily smooth. It has these folds in it. Okay, so that's the first modification. And then if you look at these folds, you have these little finger-like projections that are found on these folds that are called villi. That's the second one. And that's what we're taking a look at right here. These are all individual villi. And this figure right here should make sense. This is the mucosal layer of the small intestine right there. And so the mucosal layer is lined by epithelium. What type of epithelium is that? That's simple columnar epithelium. And then it has mucosa. Sorry. It has, not mucosa, it is the mucosal layer. It has areolar connective tissue located right below it. So that's the mucosal layer there. But the third modification is if we look at one of these simple columnar cells here and take a look at it, you have what are called these microvilli, which are these small extensions of the plasma membrane of these individual simple columnar cells. And those are going to be very important as well. just to give you an idea of what's going to happen with regards to nutrient absorption, let's say we have an amino acid right there that's ready to be absorbed. That's going to be transported across this simple columnar cell, and it's going to go into the underlying areolar connective tissue. What happens at the underlying areolar connective tissue? That's where it first enters the blood supply, and you can see these capillaries there, these blood vessels that are going to take that amino acid and other nutrients up. Then it goes down here to the submucosal layer and goes where it needs to go. But back to the point here, let's look at some of these modifications that increase digestion and absorption efficiency. The first are these circular folds here. And what they do is they greatly increase the absorptive surface area of the small intestine just by having those folds there. Another thing they do is they force chyme to spiral through the lumen. So instead of chyme just rushing through there real quickly through the lumen, what it does is it gets caught in these folds and it causes it to spiral through, to kind of auger through its way through the small intestine, which it slows its movement there and it allows much more time for nutrient absorption. So those are the circular folds, and you can see those folds there in this illustration. But then if we look on top of these folds, we have these little projections called villi. And that's the next modification that we're going to look at. These villi are finger-like projections of the mucosa, which again greatly increase the absorptive surface area of the small intestine. So this right here is a circular fold, and then you have all these villi that are projecting out from it. Same thing goes right here. You have all these villi that are projecting out from these circular folds. And here you can see, it's kind of creepy actually, but you can see all these villi here. All these little projections are individual villi, and you can kind of see the circular folds making their way through like so. Yeah, so this is let me pause it just real quick, then we'll come back. Okay, so let's take a look at this histology here. This is the mucosal layer, and you can see the submucosa kind of below it. Then you get deeper to that, and you have the muscularis externa. But if we focus up here on the mucosal layer, you can see these circular folds first of all. These are circular folds, and then these circular folds have villi projecting off of them, which again greatly increase surface area. Then if you were to take a close look at these individual villi here, they're lined by simple columnar epithelium, and then on the apical side of that simple columnar epithelium are the microvilli. So these three modifications, anatomical modifications, are greatly enhancing the efficiency of digestion and absorption in the small intestine. Yeah, this is just kind of showing you, reviewing what's happening here. But again, a nutrient here gets absorbed across the simple columnar epithelium into the blood supply, and then goes down and goes into the submucosa. So the last modifications here are microvilli. And microvilli are densely packed cytoplasmic extensions of these simple columnar cells here. So they're actually part of the cell. Here's the apical side of the cell of this simple columnar epithelium, and the plasma membrane undergoes these extensions here. And what this does is it kind of gives the mucosal surface a fuzzy appearance, and they call this fuzzy appearance the brush border. This is a really amplified, magnified view of the brush border, but when you're farther away under light microscopy, it's just kind of this brushy border there, and we'll be able to see that. But what these microvilli do is they release enzymes, and these enzymes are referred to brush border enzymes. So the microvilli, these cells here, are going to make enzymes, and these microvilli are going to secrete enzymes. So you have these brush border enzymes that are located in the lumen there, up here in the microvilli. And these are going to be important. These are going to be responsible for completing the digestion of carbohydrates and proteins in the small intestine. And here we go. This is more like we're going to see. This is small intestine histology. This is at 640 times. We can only get up to 400 max in this class, so we won't be able to see it as well. But you know, here's the mucosa. This is the areolar connective tissue underneath it, although it doesn't necessarily look like it in this picture. And then these are the individual simple columnar cells. And here we have the lumen. But if you look closely, you can see those microvilli. Okay, this little line right there, those are microvilli that are producing those brush border enzymes. And you can see from this view, it looks a little bit more hazy, which is why they call it this brush border. It's just kind of a fuzzy brush border. All right, so now let's talk about the pancreas. What the pancreas is, this is an accessory organ. First of all, it's not part of the alimentary canal itself, but it's a soft, kind of tadpole-shaped gland that extends across the abdomen here. And like I mentioned earlier, it has its head in the C of the duodenum, and then the tail over here is next to the spleen. But the pancreas does look like glandular tissue. It looks like something that would secrete something. So this is from a cadaver, but this is the glandular tissue right there. And the more you look at this kind of stuff, you can say, okay, yeah, that does look like glandular. It looks like it's going to secrete products. Okay, so the digestive function of the pancreas is to produce alkaline pancreatic juice containing digestive enzymes. And this alkaline digestive juice containing pancreatic enzymes is released into this pancreatic duct, and then it makes its way, it has to go through a sphincter here, makes its way into the duodenum, where it's going to break down all categories of foodstuffs. So I have a question for you. Why is this pancreatic juice, why is it alkaline? Why does it have a basic pH, a pH greater than 7? And the answer to that has to do with the chyme that's coming in from the stomach here. Okay, what's going on with the pH of the stomach? Super low, down here about 1 or 2. So you have this chyme, this extremely acidic chyme that's coming into the duodenum. And the stomach is kind of unique. The enzymes in the stomach, they can work at a pH 2. Not the case for pretty much all the other enzymes of the body. They have to work at a pH at about 7, including all these digestive enzymes that are going to be located in the duodenum that are secreted from the pancreas here. So the pancreas secretes these digestive enzymes into the duodenum, and for them to do their job, they have to be at about pH 7. They just don't work at that lower pH. So the reason this pancreatic juice is alkaline is because it neutralizes that pH. It takes it from about 2 and it brings it back up to where it needs to be. I want to point this out here. This is kind of a figure of what's going on within the pancreas, but these secretions that the pancreas are doing, these are exocrine secretions. And what exocrine secretions are, if you remember, these are secretions that are released onto a body surface or into an open body cavity, any space that has exposure. And by releasing these digestive enzymes into the duodenum, guess what? This is an open body cavity. And so these are exocrine secretions by the pancreas. Now the pancreas does have an endocrine function, releasing stuff into the blood supply, and that has to do with these cells right there producing insulin and glucagon, but we'll talk about that for the next section. So, but if we look here, these cells right there, what shape are those cells? What type of epithelial cells are those? Those are simple cuboidal cells. And simple cuboidal cells like to make things. They like to secrete things. And so they're going to secrete alkaline digestive juice with digestive enzymes into this duct, which is going to make its way to the pancreatic duct, and the pancreatic duct delivers those products to the duodenum. All right. So now what I want to do here is look at the different categories of nutrients and talk about their final digestion and absorption, because that happens in the small intestine here. And I want to start with carbohydrates. These are a great overview of, putting together all the different parts of the digestive tract and how they work together to break down and absorb different nutrients, which is the goal. So let's start with carbohydrate digestion. Where does carbohydrate digestion begin? It begins up here in the mouth. So here we have the way this is organized. This is the food stuff, the nutrient that we're dealing with. These are the enzymes that are going to break them down and where they come from. And this is where the site of where that breakdown is actually going to occur. So this is going to, the first place that digestion of carbohydrates is going to occur is via the mouth. That's going to occur via salivary amylase produced by the salivary glands, secreted into the salivary ducts, which make their way into the mouth. And that does is it breaks down starch. It breaks down starch molecules into disaccharide molecules. So you have these long, complex starch molecules, and it breaks it down into disaccharides, cleaves off these disaccharides there. So that's a great place to start up there in the mouth. Goes through the pharynx and esophagus, nothing. Does anything happen with regards to carbohydrates in the stomach? Nope. The next place it happens is in the small intestine. And what happens is the pancreas releases an enzyme called pancreatic amylase, which travels down the pancreatic duct into the small intestine. And that pancreatic amylase digests carbohydrates. So what pancreatic amylase does is it breaks starch down into disaccharides, but also what are called oligosaccharides, which are, maybe those have like 10 carbohydrates in a row. The point being is that pancreatic amylase takes these large starch molecules and it breaks it down into smaller molecules, into oligosaccharides and disaccharides. But we're not done yet, because the intestine, to absorb carbohydrates, we have to break those all the way down to monosaccharides. And how are we going to do this? Okay, we have the small intestine where we have pancreatic amylase in, but the final product, the final step, breaking these disaccharides down into individual monosaccharides, is also going to occur in the small intestine, but it's going to occur via brush border enzymes. Those enzymes that are secreted on the microvilli here that form that brush border. And these are those specific enzymes right there. I'm not concerned that you guys know these names specifically, but more so that the brush border enzymes are doing it. And what they're doing is they're taking these disaccharides here, these are disaccharides, and breaking it down into individual monosaccharides. Boom. And this is a great picture of that right here. Here's the lumen of the intestine. Here's a starch molecule. So we have pancreatic amylase, this enzyme, which takes this starch molecule and breaks it down into disaccharides. We're not done yet though. We need those brush border enzymes. So here we have our microvilli, And here we have brush border enzymes that are located in the lumen, but they're right there on top of the microvilli. What happens? Those disaccharides come into contact with these brush border enzymes, which break the disaccharides down into individual monosaccharides. And this is the form that carbohydrates can be absorbed from. So we have a monosaccharide, it's transported across the apical side of the simple columnar cell via active transport here. Or maybe that's, well, it's secondary active transport, but it requires a transporter to take that monosaccharide and move it into the apical side of the simple columnar cell. Then it diffuses across the cell, and another transport carrier takes that monosaccharide, moves it into the areolar tissue underneath, and then that allows that molecule to enter into the capillary and to go into the blood supply. And this does require ATP. It requires ATP indirectly to move these molecules across the simple columnar cell. What about proteins? Where does protein digestion begin? In the mouth? Nope. In the esophagus? Nope. It starts in the stomach. And as you guys know, the enzyme that breaks that down is the protease called pepsin. Okay, and this has to happen in the presence of hydrochloric acid. So we have pepsin that takes these chains of amino acids and starts breaking them down into smaller groups of amino acid. So we have a large protein, and that pepsin breaks it down into a smaller protein. Those smaller proteins we're just calling polypeptides here. The nomenclature with regards to these small peptides is kind of confusing, but just know that we have a regular long protein, and pepsin breaks it down into smaller proteins, which we call these polypeptides here. Then what? Those proteins, those large polypeptides, move next into the small intestine. And that's where we have pancreatic enzymes that are going to release through the pancreatic duct into the duodenum, that are going to take these large polypeptides and break them down into smaller proteins, into small polypeptides. And don't be worried about these pancreatic proteases there, but they are, just know that they are produced. Pancreatic proteases are produced by the pancreatic cells and digest proteins in the small intestine. But we're not done yet, because we have small polypeptides here, and for absorption to occur, we need to break it down into individual amino acids, although it does say that some dipeptides and tripeptides can be absorbed, but we want to break it down into amino acids. And again, this is where these brush border enzymes come into play. And I really like these figures here. It shows what's going on here. But here we have a string of amino acids. There we have our linear protein chain. And here you can see pancreatic proteases are coming down, and they're breaking off individual amino acids. And then we also have these brush border enzymes here that are also breaking off individual amino acids. Then what happens, that allows those amino acids to be taken across the simple columnar cell, transported out the basal side, and then enter the capillaries, go into the bloodstream, and now we have amino acids in the bloodstream to build proteins, do all that kind of stuff. Okay, let's talk about the liver here now. The digestive function of the liver does a whole bunch of things, but the digestive function of the liver is to produce bile. And like I said earlier, it's going to produce that bile, travel down the bile duct, and it's going to make its way into the duodenum. Now, bile is what we call a lipid emulsifier. This is a large lipid droplet here, a large fat droplet, and what emulsifiers do is they break this large lipid droplet up into smaller lipid droplets. Okay? And what I want to point out is that bile does not, let's see, bile itself, I'll just read down here, bile itself does not chemically digest lipids. So when we talk about the chemical digestion of lipids, we're talking about taking these glycerol and these free fatty acids from a triglyceride molecule and breaking them down into these individual parts. Okay, these individual parts here. Bile does not do that. What bile does is it takes these large lipid droplets and it breaks it down into smaller lipid droplets. So there's still intact triglycerides within these lipid droplets here. So if it doesn't chemically digest anything, then what does it do? What's the purpose of breaking it down into these smaller lipid molecules? And the answer to that is that by breaking lipids down into smaller molecules, it makes them much more accessible to digestive enzymes that are going to actually break down those triglycerides. So it's required for the efficient digestion of lipids and triglycerides. So here you can see what's going on here. You have this lipid droplet with a whole bunch of triglycerides in there, intact triglycerides, and bile breaks the lipid droplets up. So you have these bile salts that surround the lipids and are able to emulsify it. And what that does is it increases the surface area for these lipases to come in and then actually take those triglycerides and break them down into their component parts. Okay, so I mentioned this earlier, but bile that's produced by the liver cells up here can flow down into the bile duct, into the duodenum, but if this sphincter is closed here, if there's not any need for bile to be in the duodenum, then it gets backed up and it gets stored in the gallbladder. And we'll talk about what causes the gallbladder to contract. Okay, so we kind of talked about the hormones in the stomach that kind of regulate the release of gastric juice and whatnot. Well, there's a couple hormones that are released by simple columnar epithelium lining the small intestine that are also involved in this digestive process. And in particular, these hormones are called CCK and secretin. And let's see. And these two hormones, like I said, they're produced by these simple columnar cells lining the epithelium of the small intestine. What causes their release? Well, proteins and fats that are located in the small intestine are going to cause the release of CCK. The presence of acidic chyme in the small intestine is going to cause the release of secretin. So we have these food particles and we have this chyme in the small intestine, and that's going to cause the release of these two hormones. And these two hormones are going to cause an appropriate response because of it. So what you can see here is these hormones, CCK and secretin, are released into the blood. They're released into the blood, they travel through the blood, and they act on the pancreas. Guess what they cause the pancreas to do? They cause the pancreas to release that alkaline pancreatic juice that contains digestive enzymes. So this makes sense, right? You have material that needs to be broken down into the small intestine. Hormones are released in response to that. Those hormones travel to the pancreas and cause it to release the very pancreatic juice that's going to cause the digestion of that material in the small intestine. Some other things that happens, the CCK is going to travel around here to the gallbladder, and it's going to cause the gallbladder to contract, which releases bile into the small intestine as well to help with that digestive efficiency. Lastly, CCK causes this sphincter here. There's different names for this sphincter. They call it the hepatopancreatic sphincter. They cause this sphincter here to relax. And what that does is it actually allows bile, allows digestive juices from the pancreatic duct to enter the small intestine here. And this is kind of another diagram just showing this kind of stuff. Food in the stomach comes down. Now we have chyne in the duodenum, which causes the release of secretin and CCK, which acts on the pancreas, causes the pancreas to release these digestive enzymes into the small intestine. CCK also here comes back, causes the gallbladder to contract, which causes bile to go into the small intestine. Okay, so let's break it down for fat, see what happens for fat digestion. So the first place that fat digestion begins is in the small intestine. And this actually doesn't necessarily count as fat digestion, but this is where we get that emulsification of fat droplets by bile. So we have unemulsified fats that come into the small intestine. The liver and gallbladder release bile, and that causes these large fat droplets to be divided into individual small little lipid droplets. And what that allows is it allows digestive enzymes, again produced by the pancreas, secreted into the duodenum, to actually break down those triglyceride molecules. And those are called lipases. Lipid digesting enzymes are called lipases. And what are those lipases going to do? They're going to take this triglyceride molecule and they're going to break it into the individual fatty acids, there, and it's also going to separate the glycerol molecule to it. So we break down these triglycerides. We digest these triglycerides into these component parts here, which is all great. But then the absorption of triglycerides, the absorption of these parts is kind of complicated. I don't fully understand it, but I'll just kind of give you an idea of what happens here. So here we have a fat globule, and that's being emulsified by these bile salts. So there you can see the emulsification part of it. Then you have these pancreatic enzymes, these lipases that come in, and they break it down into the individual component parts. The next thing that happens, they form this thing called a micelle, which I'm not too clear about, and that allows that material to diffuse across the simple columnar cell into the underlying areolar tissue right there. And instead of these different component parts entering the blood supply, they actually enter into the lymphatic system. So remember, the lymphatic system drains this mucosa, and what it does is it takes up these glycerol molecules and these free fatty acids. And then it enters into the lymphatic system. And this is all the lymphatic system right there. This is a detailed thing of what's going on here, and it's kind of to help me with the whole thing. But here we have the small intestine, and here we have dietary fat, and the pancreas releases lipases, and it breaks those down into free fatty acids and monoglycerides. Those travel across the simple columnar epithelial cell here, and now they get into the lymphatic system. Let's see, that's the bloodstream they're showing there. So eventually they go from the lymphatic system and they move into the bloodstream. Okay, so they go from the lymphatic system into the bloodstream, and then those different component parts get delivered to the different parts of the body. that they need to. So for instance, here's a fat cell, and that's going to take up these component parts and put them back into triglycerides, and so now we have a fat cell that's storing triglycerides. These are also going to be taken up by different cells of the body. This is a muscle cell, and it's going to be used for energy. Okay, so I want to bring the liver in back into this again, because the liver plays a bunch of different critical roles here. But the point that I want to make here is that all blood leaving the absorptive surface areas of the digestive tract enter into what's called the hepatic portal system, and it flows to the liver. So what this is representing here, these are just different component parts of the digestive tract. Stomach, this represents the small intestine, these are all the large intestine, and these are, you can see the blood vessels that are draining the digestive surfaces. So they're taking these nutrients, we'll do here in the small intestine, they're taking these nutrients, they're absorbed into the blood, and the question is, where do they go next? What happens is all these digestive surfaces are drained, and they merge into a vessel called the hepatic portal vein. That's the hepatic portal vein right there. And what it does is it takes this nutrient-rich blood and it takes it to the liver. So now the liver has first shot at all this nutrient-rich blood, and it can do different things of it just depending on what the situation is in the body. So I guess I didn't put this on here, but like for instance, let's talk about glucose. Let's say glucose, individual monosaccharide here in the small intestine. It gets absorbed into this vessel system, the hepatic portal system, and it eventually makes its way to the hepatic portal vein right there. And now that this glucose molecule goes to the liver. What's the liver going to do with that glucose molecule if blood sugar is super high? If you have hyperglycemia? What's it going to do with that individual glucose unit? And I'll let you guys think about that. I know it's on the study questions. Here's some other stuff about the liver that you can read about. One of the things the liver is also involved in is it's the body's detox center. It takes a lot of chemicals and breaks them down into non-toxic forms. And that's what I want to point out here. So this is kind of separate from digestion, although we have the potential to receive toxins through the GI tract. So let's see, there's three main ways that toxins can enter the body. Through the lungs, we can breathe them in. They can be absorbed through the skin, and they can go through the GI tract. Okay, the GI tract is really precarious. So what those toxins do is they make their way to the liver, and that's where the liver detoxifies those. It takes those toxins and it breaks them down into non-toxic forms. And then, we want to get rid of these parts. So how can we get rid of these parts? Well, they can exit the body through the skin. We can sweat them out and whatnot. They can exit through the kidneys, more specifically through the urine. They can go out through the colon, more specifically through the feces, or they can go out through the lungs. We can breathe them out. So kind of some side notes there about the liver, just because the liver does so many things. But that's good.